Hello everyone, my name is Maria Mendoza and this is my first time recording one of these lectures, so please bear with me. This PowerPoint is going to be on chymotrypsin and I put it together using your textbook. So these are the figures and then I went ahead and incorporated some information that I found in the paragraphs. So what is chymotrypsin? Chymotrypsin is a protease enzyme that catalyzes the hydrolytic cleavage of peptide bonds. It doesn't hydrolyze the peptide bond directly. Instead, the process happens in two phases. It is specific for peptide bonds that are next to the aromatic amino acids, tryptophan, phenylalanine, and tyrosine. It enhances the rate of peptide bond hydrolysis by a factor of 10 to the 9th. It also catalyzes the hydrolysis of small esters and amides. What happens in the chymotrypsin mechanism? The two phases are acylation and deacylation of a serine residue. Figure A shows the primary structure of chymotrypsin. It is made up of 3 polypeptide chains, chain A, chain B, and chain C. The chains are linked to each other via disulfide bonds. The amino acids in red are important for catalysis. This figure shows the surface of chymotrypsin. Yellow is equal to the hydrophobic pocket. This is where the aromatic amino acid side chain of the substrate will end. Remember, the substrate here is whatever polypeptide or protein is going to be cleaved. The red part is the active site residues, serine, histidine, and aspartic acid, or aspartate. In figure C is a ribbon structure of the polypeptide backbone. The disulfide bonds are shown in yellow. So here you see the tiny A, the B, and the C chain. Figure D shows the active site with a bound substrate in white and yellow. So this is the substrate right here. The oxygens are red. 1. The hydroxyl of serine 195 attacks the carbonyl group of the substrate. Oxygen begins to develop a negative charge, which is stabilized by the oxyanion hole, the amide nitrogen from serine and glycine in blue. The substrate aromatic side chain in yellow sits in the hydrophobic pocket. The amide nitrogen of the bond that is getting cleaved is in white. Figure 6-21 is an example of chymotrypsin catalyzing a reaction. So the rate is measured by the release of p-nitrophenol from p-nitrophenylacetate. So this right here is an ester group, and let's see, this is an acetyl group. So in phase 1, this is the fast acetylation phase. The peptide bond is cleaved, and an ester linkage is formed between the carbonyl carbon from the peptide and the enzyme. An acetyl group is added from the substrate to the enzyme. The result is a covalent acyl enzyme intermediate. So all of the words that are in bold, you should know those words. So an acetyl group is added from the substrate to the enzyme. The result is a covalent acyl enzyme intermediate. In phase 2, this is the slow deacylation step. Here the ester linkage is hydrolyzed. That's when water is added and the bond is broken. You are left with an enzyme with no acetyl group, so you're left with a free enzyme. So this is figure 6-4. Wait, 6-23. And I think it's 6-23. Okay, so the hydrolytic cleavage of a peptide bond by chymotrypsin. So this is the chymotrypsin free enzyme. This is the active site, and you should know it very well. You need to know what these components are. So this is the hydrophobic pocket. This is the oxyanion hole. And then we have serine, histidine, and aspartate. Aspartate, histidine, and serine make up the catalytic triad. The catalytic triad helps to cleave peptide bonds. Again, that's serine, histidine, and aspartate. We also have glycine, but glycine serves a purpose later on in the pathway. The purpose of glycine 193 is to stabilize the tetrahedral intermediate formation, transition state, and breakdown, transition state. This is an example of the use of binding energy through enzyme transition state complementarity. And this glycine is on the backbone of chymotrypsin. Don't forget, we also have the oxyanion hole and the hydrophobic pocket. It's also important to note that chymotrypsin is specific for peptide bonds that are next to the aromatic amino acids, like tryptophan, phenylalanine, and tyrosine. Here we have phenylalanine right here. And this is the backbone of the polypeptide that serves as the substrate. You have the NCC, NCC. So the bond that is actually targeted by chymotrypsin is actually this bond right here between this carbonyl group and this nitrogen. So when the substrate binds, the aromatic side chain nestles in the hydrophobic pocket. This positions the peptide bond so that it can be attacked. So here you can see that the substrate is now nestled inside of the active site of chymotrypsin. So this blue part here is right here. And then we have the aromatic side chain inside of the hydrophobic pocket. The carbonyl is actually inside of the oxyanion hole, and then we have the rest of it here. So aspartate will stabilize histidine via a low barrier hydrogen bond. So remember, aspartate is over here. It's going to stabilize histidine via a low barrier hydrogen bond. Histidine becomes a stronger base and it deprotonates serine. So this is the oxygen, the hydroxyl from serine, and histidine deprotonates it. Serine then becomes a strongly nucleophilic alkoxide ion. The oxygen ion on serine then attacks the carbonyl group of the substrate. So it is attacking this carbonyl right here, the one that's next to the aromatic side chain. So the oxygen ion on serine attacks the carbonyl group of the substrate. At this point, a tetrahedral acyl enzyme intermediate is formed, and it gives the carbonyl oxygen a negative charge. This negative charge is short-lived, and it is stabilized via hydrogen bonding in the anion hole right here. Okay, so this is step 2. The negative charge on the carbonyl that was being stabilized via hydrogen bonding in the anion hole is unstable, and this instability leads to collapse of the tetrahedral intermediate. At this point, a double bond with carbon reforms. So your carbonyl reforms, and this displaces the amino group of the peptide linkage. That's this guy right here. Where am I? This action breaks the double bond. The fact that histidine is protonating the amino leaving group makes the displacement easier. Okay, sorry, I think I have to fix this. So at this point, a double bond with carbon reforms, and this displaces the amino group of the peptide linkage. This action breaks the bond, so not the double bond, but just the regular bond. This bond right here, the peptide bond. The fact that histidine is protonating the amino leaving group makes the displacement easier. So that's right here. Histidine is protonating the amino group that is leaving, and it's easier to displace. Here's step 3. So you can see that the leaving group is gone. Once the peptide bond is broken and the leaving group is gone, an incoming water molecule is deprotonated by general base catalysis, resulting in a strong nucleophilic hydroxide ion. This hydroxide ion then attacks the ester linkage of the acyl enzyme. You can see the attack right here, resulting in a second tetrahedral intermediate. An oxygen is left and the oxyanion hole. Oh, yeah. Again, that oxygen will have a negative charge right here. The second tetrahedral intermediate then collapses and forms the second product, a carboxylate anion, which displaces the serine. The second product dissociates and leaves. This regenerates the free enzyme. So this is the second product. In summary, this process has 2 phases, the acylation and deacylation phases. In step 1 through 4, this is the acylation phase, and the formation of a covalent acyl enzyme intermediate is coupled to the cleavage of a peptide bond. In steps 5 through 7, this is the deacylation phase, which regenerates the free enzyme. So here's some extra notes. The effects of pH on chymotrypsin activity. As you might remember, protons on the different side chains are lost at different pHs. What are the individual contributions of Kcat and Km? Again, this is the enzyme turnover number, Kcat, and Km is the substrate concentration at half of the maximum velocity. So at low pH, histidine 57 is protonated. Remember that the pKa for histidine is about 6. Therefore, histidine loses its proton at a pH of 6. If the pH is lower than 6, then that means that histidine is protonated. If histidine is protonated, this means that it will not remove the proton from serine 195 in step 2. This will result in an overall decline in the enzyme turnover number, or the Kcat. So with high pH, under normal pH, the alpha amino group of isoleucine 16 forms a salt bridge to aspartate 194. This salt bridge stabilizes the active conformation of the enzyme. However, at high pH, this amino group loses its proton, so the salt bridge never forms. What happens instead is that the hydrophobic pocket will close. If the hydrophobic pocket is closed, the aromatic amino acid won't have a place to be inserted. This will result in an overall increase in Km. An increase in Km means that you will need a higher substrate concentration to reach the maximum velocity.